Good afternoon, ladies and gentlemen. My name is Midshipman Lillian Usadi, and I'm a student at the United States Naval Academy under the advisors Lieutenant Stephen Yi, Dr. Hatem El-Bidwehi, and Dr. Samara Firebraw. Today I'll be presenting on the manipulation of microrobots using Chladni plates and multimode membrane resonators. Just as a broad overview, micro/nanorobots have tremendous potential in the physical, chemical, and biological domains. And the purpose of this project is exploring the use of acoustics, specifically Chladni plates, to move these microrobots around as a source of actuation. I'll be going more into details of this experiment throughout this presentation, so I will move on right now. So the motivation, microrobots, which often are 10 to 100 micrometers, are able to interact with biological structures on the same size scale that macroscale robots cannot. They have awesome and fantastic applications, which include many in biomedical, the biomedical domain, including lab on a chip, which is a 2D sort of application, drug delivery, tissue repair. There are many ways to manipulate microrobots, including optical ways using lasers, chemical, electrical, magnetic using inductors, and in this case, acoustics as well. Acoustic actuation is a non-invasive way to manipulate microrobots. This is in contrast to some optical means, such as lasers, which have the potential to burn the tissue with which you're operating in, as well as chemical, which could change the chemical composition of the medium with which you're using it in. Acoustics is a very good way that is non-invasive. Chladni plates are thin metal plates that have natural resonant frequencies withstanding wave patterns, visualized by particles displaced in the plate. Chladni figures are complex patterns formed in response to vibrations, and we can see one on the figure 3 as well as a slew of them in figure 4. In figure 3, we can see a Chladni figure on top of this Chladni plate. Underneath the Chladni plate is a piezoelectric actuator, which oscillates at various frequencies, and at certain frequencies, the eigenfrequencies, or better known as the resonant frequencies, we get the Chladni figures. The sand will often conform to the nodes, the places of least movement, and move away from the antinodes, the places of most movement. Sometimes the opposite phenomenon happens where the particles move to the antinodes and away from the nodes, which is an interesting phenomenon, but only happens sometimes. The idea is to make displacement maps, which show the direction of movement of a particle at every single point on the Chladni plate, as a result of plate oscillation at the indicated frequency. And then later on, I'll be able to use these displacement maps, and they're referenced by a control algorithm to control the movement of a microrobot or other particles. So I want to be able to harness these interesting patterns, these Chladni figures, and model them and how they make the particles move on top of them. The figure 6, you can see a notional schematic that doesn't necessarily align with anything that actually happens, but an idea of what I would want to do. You can see the blue bead is a stand-in for a microrobot and would move to the left if I apply 2.5 kilohertz, and then after some time, 0.75 seconds, I would apply 3.4 kilohertz, and then the bead would move to the right because the displacement map changes. And ultimately, the bead would arrive at the red X, which, if you only had played 2.5 kilohertz, you would not be able to reach the destination that you intended. This experiment has two parts: the macroscale manipulation in air, which is primarily a replication of previous experiments, and then microscale manipulation in water, which is the novel part of the project. Table 1 shows the two different phases. You can see that we start out with using 750 micrometer solder balls using a frequency around 1 to 20 kilohertz range, and then we will have to take everything that we learned in that first phase of the experiment and apply it to the second phase, which is the microscale manipulation with microscale membranes, and we'll be shrinking everything down and even be using microbeads, 50 micrometers, as well as much higher frequencies. So now I'll talk about the first phase of the experiment, the macroscale manipulation with Chladni plates in air. Here's a picture of the macroscale experiment. Random frequencies from figure 4 were played via the MATLAB controlled signal generator to drive a piezoelectric actuator, and a 50mm square Chladni plate. And then the images were acquired concurrently using a USB microscope. On the right, you can see we have my computer, which is connected to the signal generator, then fed through an amplifier, then fed to the Chladni plate, and then ultimately pictures were taken with the microscope. In figure 10, we can see one of the Chladni figures formed at 3.951 kilohertz. In this picture, I was using sand particles before later on I moved on to soldered beads. And ultimately what I got from this stage of the experiment was the ability to identify the particles as well as make displacement maps. On row A, we can see the different Chladni figures that were formed as a result of the 6 frequencies that were played. Then the computer was able to identify, and I was able to identify the particles on these plates, which is row B. And then row C, I was able to map the movement of these particles effectively. And we can see that it's quite clear for example, 7.902 kilohertz, that the displacement map looks quite similar to both the object identified particles and grayscale images. There still needs to be some work done with the algorithms, but we thought that it was good for now and moved on to the second part of the experiment. So the second part is the microscale manipulation with microscale membranes. So here's the microscale experimental setup. It's quite similar to the macroscale setup. Frequencies to drive the piezoelectric actuator and multimode membrane resonators were chosen based off of a previous paper as well as prior analytical computations based off the chip size, using the use of water, the use of no water, and the images were also acquired concurrently using a USB microscope. One thing to note, we also applied an insulating varnish layer on top of the entire chip because we would be moving to water and I didn't want to short anything and also destroy the chip as well. And also there is an air bubble in between the microchip and the piezoelectric actuator, similar to the reason why a drum head needs some air in between the top membrane and with the table or whatever the drum head is on. You need a space to resonate. Figure 13 shows a bigger picture, an actual picture of the experimental setup. Here we have a closer look of the microchip. The 10 micrometer thick silicon microscale membranes were designed in-house and then manufactured by an outside microchip manufacturer called Memscap. Each chip quadrant contained different shaped membranes and boundary conditions. Figure 14 shows a more zoomed in picture of, you can see the microchip on the package and the power and ground. And then figure 15 shows an even more close up image with the microchip on top of the piezoelectric actuator on top of a package, and as well as the power wire glued to the piezoelectric actuator. Now we're going to go through the different quadrants and the reasoning for each quadrant and the design decisions that I made. So one quadrant we're going to call the 2D drum head clamped. So it's just mostly square, so different sizes, clamped on all edges. The left picture is the design and the right picture is an actual photograph. The actual photograph, you can't see anything because the drum head is clamped and so there's no space within chip and you just see the top layer of the membrane. Now we have the same design except we're going to call it the 2D trampoline and it's unclamped. So it's almost the same, but in this case the sides are mostly free. And on the right hand side, this picture, you can see the outline of the membranes because you can see basically the light streaming through the holes of these membranes. Then we wanted to go on to this 1D beam idea, and we had the unclamped and clamped. On the left, you can see the clamped 1D rectangular shapes of different heights and different widths. And on the right, we can only see the unclamped 1D beams, which, as I explained before, 2 slides before this, the reason for that is that we can see the light streaming through the little gaps in these membranes. And then lastly, the fourth quadrant has a mixture of all of the 3 other quadrants: the 2D drumhead clamped, the 2D trampoline unclamped, and the 1D beamed unclamped and clamped. Now I'm going to talk about the imaging and microparticle detection part of our research. Microparticle detection techniques used on the macroscale are effective on the microscale. In this case, in figure 20, we can see that the microparticles, 50 micrometers, are imaged on the 2D drumhead clamp chip at 890 kilohertz. On the left, you can see frame 1, which is the beginning, before the note is played, and then frame 50, after the note has been played and is turned off. Clearly, the beats conform to different beads and bunches as a result of the resonant frequencies. And the second row of images shows effectively identified particles using the post-image processing algorithms. And after that, I was able to ultimately model and make displacement maps for the particles on the microscale. And it is proven that the map algorithms used on the macroscale is effective on the microscale. I did this for 890 kilohertz, the frequency that we explored in the previous slide, as well as for 440 kilohertz. As well, there are a slew of other frequencies that also work, but I am just showing 2 for now. I was then able to overlay the displacement maps with the original image on the microscale, as well as the microchip designs. I know it's a bit hard to see, but we can see that the beads do conform to the different membranes, and we can see that there's clearly resonant frequencies and an interesting phenomenon happening on this microscale. Ultimately, the microscale experiments require new displacement maps due to the differing physics in resonators, but the process is similar and informed by the macroscale results. On the microscale, the effects of acoustic streaming, effective weight forces, all these phenomena as a result of the microfluidics severely affect the movement of the particles. For the future, there needs to be work done in implementing control algorithms for these displacement maps, as well as improving the displacement map algorithm itself. I would like to thank the National Science Foundation and the Trident and Bowman Scholar Programs of the United States Naval Academy, as well as my advisors, and to Micro Machines for letting me present at this conference. Thank you very much.